if you happen to be your large intestine or your small intestine, stomach, how do you know a big bear is attacking the rest of the body? Well, you don't, right? The autonomic nervous system would handle a lot of that response with your internal organs. So that's an example why you would need it. Because the last thing you need, if you're fighting for your life, is to be diverting a huge amount of your body's resources to digesting your food. It's going to do you no good at all at that point in time. So slow down the activity there, don't waste the calories, don't send a lot of extra blood there, that sort of thing. This is just a schematic. You don't have to know this thing. There's nothing extra on here. It's just visual to help you understand what they're saying. And what they're saying is that you would have sensors in the mucosa of the GI tract, and they would have sensory neurons that then go into the enteric with some other extensions that are connected to the ANS and the central nervous system. Okay? And they're just trying to show you that short of sending back information to affect these, basically this just runs itself. So some of the neurons literally just hook immediately in a loop, either going through the submucosal plexus or the bienteric plexus, so that this can just run itself. So let's just talk about what's going on in some of the organs here. In your mouth, you produce saliva. It's mostly water. We're going to see that's a common pattern, but by now you're probably used to that in the body. Has some solutes in it. Primary solute we're concerned with, though, is an enzyme called salivary amylase. Salivary amylase, we'll talk a little bit more about what it does digestively in a moment. Note that while the glands do produce the water, the solute salivary amylase, if you were to analyze in detail the chemistry of the saliva from different glands, so you have several sets of saliva producing glands, it's not identical. Okay, there are differences. We're not going to get into that because they're all pretty minor. But you need to be aware that it's not full redundancy. So if something happened with one salivary gland and then you lose that salivary gland, doesn't mean it's all going to be okay, because the other one's not necessarily constant. All right, what controls its production? It's under autonomic nervous system control. So note that when we're up in the mouth and up in the upper parts of the pharynx at least, there's no enteric yet. The enteric doesn't really kick in until you get to the esophagus. So we're still under autonomic nervous system control, and parasympathetic control will make you produce more saliva, sympathetic stimulation less. Okay? Last thing you need to be doing when you're running from the bear is drooling on yourself. That's basically what we're looking at. Your tongue, you have one that's in the mouth. Basically, that's it. Other than it is important for maneuvering food, and it does produce an enzyme called lingual aromatase. We'll talk more about that here in just a moment. Now, so that's the things that they're doing, some basic stuff dealing with the mouth. It turns out, now we can start talking about digestion, and there's a couple of key terms you need to be careful with here. Digestion means to break something down into smaller components. Okay? Don't confuse that with another term we'll mention shortly called absorption, which is bringing materials into the body. Sometimes students will get that confused. The other thing is that there's two terms related to digestion. That's mechanical digestion and chemical digestion. Mechanical means physically breaking food into smaller pieces. Okay. Chemical means changing the chemical bonds in the food. In other words, changing it from one molecule to a different molecule. In the mouth, chemical digestion is chewing or mastication. Okay, please don't confuse that with a similar sounding term. You don't sound silly. Okay. So chewing or mastication, and what you do is you chew it up, break it down until you can form it with the tongue into a little ball. Okay, that actual structure is called a bolus. Okay, so when you swallow a bolus of food. Chemical digestion, what's happening is we told you had salivary amylase, and what it does is it breaks down starches. Okay. Note, and we're going to come back to this a little later, the only carbohydrates we can absorb into the body are monosaccharides, simple sugars. You can't do disaccharides, trisaccharides, complex carbohydrates, polysaccharides, can't do any of that. It has to be monosaccharide. Okay? The problem with your salivary amylase is that it cannot tolerate an acidic environment. So as soon as it hits the stomach, it gets broken down and becomes inactive. So the only digestion it really gets to do is while you're chewing your food and in the short trip of the food down the esophagus. The lingual lipase works on triglycerides. Okay, so it breaks those down. It's a little different though than your salivary amylase. You see, you chew it with your food in the mouth, but it doesn't work in the mouth. It turns out for it to work, it needs to be in an acidic environment. So it actually isn't activated until it gets into the stomach. Okay. Mm-hmm.
Yeah, in the video, they posted about this, said something about food having to turn into this. It's another chemical to be able to be.
Is it in the video I made?
Yeah.
Oh yeah, so what I'm saying is, what I'm saying is you can't absorb anything but monosaccharides. So any of the other things you eat, like a starch, which is a complex carbohydrate, has to be broken down into individual glucose molecules. Otherwise you can't absorb it.
Okay.
Alright. So this is a pretty good example of something I want you to know for the test. This slideshow, parts of this are a departure from the usual rule I have in the class about know everything on the slide, right? We're using that, I'm telling you that. Here, not so much. What I need you to know whenever you see one of these enzymes, and enzymes normally end in ASE, ACE, that's the definition for an enzyme. I don't need to know the details of its digestion, what it's breaking down into what in detail. What I need to know is it digesting one of the four main molecule groups, right, and which one? So in other words, is that enzyme going to digest proteins? Is it going to digest lipids? Is it going to digest carbohydrates? Or is it going to digest nucleic acids? Okay? That's what I need to know. So on the test, I would not say does salivary amylase act on starches or on glycogen or a whole bunch of other stuff. You would just need to know it breaks down carbohydrates. That's it. All right. I don't need you to know lingual lipase works on triglycerides. I need you to know it's a lipid digesting enzyme. That's good enough. Alright, so turns out when you swallow your food, it goes into the pharynx and the esophagus. Turns out they're not overly exciting in your whole overall chemical sense. They don't produce enzymes or absorb anything, they're just transport. Okay. nothing much going on physiologically there. However, the swallowing is more complicated than you might think. There's 3 different stages of it. The first is called the voluntary swallowing stage. Note that the technical term for swallowing is deglutition. Okay? So voluntary deglutition is when you push the bolus back behind your tongue. So what you think of as swallowing. As soon as it goes back into the pharynx, then the involuntary swallowing kicks in. And in this case, it's going to just move it into the esophagus. So that's called pharyngeal deglutition or swallowing. And once it gets into the esophagus, that's the esophageal deglutition or swallowing. Involuntary passage all the way to the stomach. And the esophagus is where we engage what we call peristalsis. Peristalsis is the controlled muscle contractions that move the food through the GI tract. Okay? So it pretty much remains the same from here all the way through the whole system and out the anus. All right? So it's all coordinated muscle contractions, either churning your food, mixing your food, or moving it along. Now, once we get down that esophagus and we make it into the stomach, things become a little more interesting. We have a little bit more going on. First of all, for digestion, basically your stomach just mixes or churns the food. It's not really trying to crush anything or whatever amount of you even. So it's just mixing it up. But it keeps doing that until it reaches a pasty consistency, and they call that material chyme. So it tries to take the food it chewed up, turn it into chyme, and the chyme is what is released into the small intestine. Now, chemically, we have a bit going on here. First of all, remember that lingual lipase that you chewed up with your food and swallowed, well, it's going to now digest triglycerides into fatty acids and diglycerides. It can do that because it's now in the acidic environment of the stomach. Note that on the test, all you need to know is the lingual lipase, once it's in the stomach, would break down lipids. You don't have to be more specific. Okay. We have some cells called parietal cells. They produce HCl, hydrochloric acid, so that's what makes your stomach acid. It's useful because it does kill many microbes. Most microbes cannot survive this, but unfortunately for us, some can. And it denatures proteins. That's the big helpful thing it does for us digestively. Remember that protein structure is very sensitive to pH. So when you have a protein and it has a certain shape, all the strands of the amino acids are being held together with bonds, primarily hydrogen bonds, which are fairly weak. When you get a lot of hydrogens around, they start breaking loose, the hydrogen bonds do. So that essentially, it's like having a big ball of yarn and then it starts unraveling, okay? That's important because that means that now the enzymes can get inside the protein and start attacking it more efficiently. Otherwise, it would only be able to contact the outside and not digest it well. So this acidic environment is important for digesting your proteins. You have some cells called G cells in the stomach. They secrete a hormone called gastrin. And what gastrin does is increase digestive activity. So it actually makes your stomach would churn more and actually produces more hydrochloric acid and other things as well. So it just increases overall digestive activity. You have a new actual enzyme here. Your stomach produces its own lipase called gastric lipase. So it digests triglycerides and fatty acids and monoglycerides. But once again, I just need you to know it digests lipids. And finally, we have some cells called chief cells, and they secrete a chemical called pepsinogen. And pepsinogen, in the acidic environment of the stomach, alters to something called pepsin. And pepsin is a digestive enzyme. So the pepsin digests proteins. Now, this slide is a really good slide for me to give you a piece of advice. Whenever you get in a situation where you're seeing a lot of repetitive stuff happening that you have to memorize, such as enzymes, and the enzymes do different things, or they're made in different places, or whatever, it's always a good idea for you to go through your notes carefully and make yourself a table, okay, that organizes this stuff. Your book, and all of them, just yours eating, will have that table already in there of some kind that'll list enzymes and what they do. They can be very useful. The only difference is that it will usually have more information than you need, right? More things that I didn't include or something, and it doesn't often put them all together. If I were you, I would make mine a table. So it would be something like, maybe you could write a column with, say, enzyme in it. So maybe the enzyme is here, and then you would write your enzyme here, and then the next thing is, or maybe you do the enzyme here, and then you go over here and you go, okay, which organ does it work in, and which organ is it made in, and all sorts of stuff. Just however you want to set it up. Because it'll do two things. It'll make you go through the notes thoroughly, so you don't miss anything, and you'll learn while you're making the table. And then once you're done, it gives you an excellent study guide, cheat sheet thing. Oh, there's one other thing. Watch for tricky stuff here, because this is why you want to make a table out. For instance, we've got our lingual lipase, and that's great. But we've got another lipase, gastric lipase. Don't confuse them. Right? They're made in different places. Right? Both digest lipids, but that's the way it is. All right, that's digestion. So what about absorption? Well, we don't absorb much through the stomach, actually. So we can get water absorption. That works out rather well. We can absorb ions easily through here. Some short chain fatty acids, in other words, your smaller fatty acids from the lipids you digest, we can get some of those in here. Drugs, most drugs will go in here, well, I should say some. I think some is more accurate. We get some drug absorption here, and alcohol will be absorbed in this stuff. But most things not. Stomach isn't good for that. Now, once we leave the stomach, we got to be really careful about something. And that is, we made it through the mouth, pharynx, esophagus, the stomach. In the GI tract, the chyme is going to go through the small intestine and large intestine next. But when we get to the small intestine, we have to start considering some of our bigger, major accessory organs. Because the liver and the gallbladder and the pancreas, all their products are carried to the beginning of the small intestine and released there. Okay? Now, that part is called the duodenum or duodenum of the small intestine, and that's where those things will work, even though the food never goes through them. So be careful that in your anatomy, if you're learning your anatomical terms and you learn the ductwork form, that'll become apparent. But you just need to make sure that you don't get that confused. So the first thing we're going to talk about is the pancreas. Pancreas produces a lot of what is called pancreatic juice. So that's the fluid it produces. It produces over 1 liter a day. It's mostly water, but it has some important things in it. One is sodium bicarbonate. Sodium bicarbonate is bicarbonate, which you learn, but you add a sodium to it. Okay? Sodium bicarbonate is a buffer. In other words, it reduces acidity. A buffer is something that reduces acidity or alkalinity. Okay? A helps make it more neutral. So it'll buffer the stomach kind, which is very acidic when it comes out. It also has a lot of digestive enzymes. In fact, it has enzymes that will digest in some manner all 4 of your macromolecule or big biological molecule types. Okay? So it has stuff to digest carbos, proteins, lipids, and nucleic acids. Liver and gallbladder, also remember 2 things that the food never passes through. They're going to produce things and put them into the duodenum of the small intestine. The liver produces bile, and the gallbladder stores and concentrates the bile. It turns out you make a lot of it, around 1 liter a day. It contains bile salts, okay? And these act as emulsifiers. Remember, salt is something that's not an acid or base. That's basically all it means. But an emulsifier works a little bit like soap. Okay, soap's basically really an emulsifier. So here's your problem. If you're eating lipids and they go into your stomach and you're dumping this acid and all this other stuff, and your enzyme, what you're dumping into your digestive tract is mostly water. Okay? And what's the deal between lipids and water? They don't dissolve. Lipids don't want to dissolve in water. So now you got a problem, because what happens is when you eat that, like when I ate all those nachos covered in cheese last night, chili on them, it's probably half fat. All right. When it gets into your digestive tract, basically the lipids all want to stick together away from the water. So you just get a big wad of lipid in you. All right. That's problematic, because now the enzymes, they can only attack the outside. All this is immune to their action, right? But what emulsifiers do, they'll actually stick to the water and to the lipid. So they help turn this into this. So that now you've got a lot of little lipid pieces, and the enzymes can attack them a lot better. All right, this stuff is extremely important for lipid digestion. Okay, these bile salts. Turns out your liver makes cholesterol as well. You know, cholesterol is one of these. To me, it's really wild, because they had just identified there being a link between cholesterol and cardiovascular disease when I started college. At that point in time, nobody cared about cholesterol. And then when they first discovered it, then it was like, oh, we're all gonna die from cholesterol, eggs are evil, you know, all this stuff. But now they've kind of changed. We now know that we have high density lipids and low density, as we discussed, and all these other things, right? It's more complicated. But what a lot of people don't realize, and they forget, and you probably studied some of this in AP1, is that you have to have cholesterol to build a cell. Cholesterol is probably the second most important component of the cell membrane. You cannot make a cell without cholesterol. Okay? So it would be impossible for you to live with zero cholesterol, because your cells will fall apart. So you have to have it. Well, how does your body get it? Because you're probably thinking, you know, now they're like, well, cholesterol's bad, it's going to kill you, so don't eat it, and everything else. And historically, humans generally had fairly low cholesterol diets, very low in fact. So how are they getting cholesterol to build cells? Well, your body makes it. And it's the liver. Your liver makes its own cholesterol. And it goes a roundabout way of getting it into you. It makes the cholesterol and puts it into the bile. The bile then goes to the digestive tract. Then you absorb the cholesterol through the lining of the GI tract to get into your blood. Okay? And that's normal. That's how you're supposed to do it. The argument, and I don't know that this has even been decided yet, because they still keep doing all these studies on it, is if you have high cholesterol, where is it coming from? It used to be we were eating too much cholesterol, and now they know that in many cases, altering diet does not change a person's cholesterol level. And it makes sense it might not, because this makes, the liver makes its own cholesterol. So if it wants to make a lot, it's gonna make a lot. So they have drugs to try and control that. Statin drugs control the absorption in through the lining of the intestine. Okay, and they have other things that actually work in the liver. All right, so we got our bile salts to emulsify, break up the lipids mechanically, not chemically. They're not altering the lipid. Okay, so it's just a mechanical digestion. The cholesterol is being produced by there, and also bilirubin is found in the bile. That is a breakdown product from your damaged red blood cells. So when they digest the red blood cells, when they break up the damaged red blood cells in the spleen and liver, it forms this bilirubin. And your liver removes the bilirubin and puts it in the bile so you can excrete it. Okay. This is actually what gives your feces a brownish color. For the most part, without this, your poop is basically the same color as what you eat. Okay. Those are really heavy magical changes when it undergoes digestion. What gives it a brownish color is primarily its bilirubin. Now, as we're going to discuss in a moment, it has been argued that the most important component in your digestive tract is the small intestine. It does a lot, and a lot of absorption happens here. And one of the reasons it's so efficient at that is it has 3 different levels of structures that increase its surface area inside. So you might think of it initially as a long tube, like a hose. But these structures actually increase the surface area many-fold. And I forget what it is, but I think it's something like 8 times or more what a regular smooth tube would be. So it has a large internal surface area. I listed them from the largest to the smallest structures, okay? So I'm going to mention them real quick, then I'm going to show you a figure. Circular folds are clearly visible. They're folds on the inside that you can see that make it irregular. Okay? They cause the chyme to spiral. They're kind of offset, and so as it's pushed through, it kind of flows around one and then hits another one, and it makes it kind of spiral as it goes through. The villi, sometimes pronounced villi, are finger-like projections in the mucosa. And each little finger, each little thing sticking up, has its own blood vessels and lymph vessels in it. Okay? Finally, you have microvilli, and these are very tiny microscopic structures. They're actually cell parts. Okay? So these are just projections of the apical membrane of the absorptive cells. In other words, the cells lining your intestine have these little finger-like things sticking up out of it. Okay? So let's take a look. I actually like this figure from the internet. So I'll use this because it puts them all there at once. I like what it's doing. So here's what we've got. Here's your small intestine. If you were to section it, see how the inside has these rings around it like that. Okay. So you can literally see those. Those are those circular ridges going around. Okay. But if you were to zoom in on the surface, it's not smooth. It actually has all these little finger-like projections sticking up from it. Now these range, you can kind of see them with an unaided eye, but not well. I couldn't, because I'm too old. They range from about like 0.5 millimeter to a little over 1 millimeter in total length, so they're really tiny. What it tends to look like in a real intestine is that it makes the intestine except for the mucus on it, it looks shiny, but the surface isn't smooth looking, like say the top of your desk. It almost has kind of a velvet look to it, and that's because it's covered in these. Okay? Note, what happens is they all have their own little lymph capillary here, and they got their own blood supply. Okay. So this is a villus, or one of the villi. Now note, they would have epidermal cells covering them. And if you zoom in on a little section of epidermal cells, you see this. Each epidermal cell has these little finger-like things like that, and those are the microvilli. Okay. When scientists first found these, and they're trying to describe all this, they thought these looked like the bristles of a brush. And they realized these were there to increase surface area, and so they said, well, you know, when you think about it, all the real action going on in your small intestine, this is where it's happening. Because if you release an enzyme from this cell, it's going to be released right here. If you've got chyme coming through here, well, it doesn't matter if it's out here because it can't be absorbed. Only the chyme that comes and makes direct contact with the surface of the cell can be absorbed. So they term the region where the microvilli are the brush border. All right? And they refer to a lot of things that are happening with digestion in the small intestine. They refer to it relative to the brush border because that's where it's really happening. One of the things that one of the times they do this, it can become confusing to you, is in this term. You see, any enzymes, since they're produced, any enzymes produced by the small intestine, since they've been working in this brush border area, they started calling them brush border enzymes. Okay. To me, that was a little confusing. I like things pretty straightforward. If we have gastric enzymes or stomach enzymes, right? We have lingual enzyme. Why don't you just call it a large intestine enzyme? But they don't. They're going to call it a brush border enzyme. So from your standpoint now on, when you hear brush border, that means large intestine. I'm sorry, small intestine. So that's what's happening. So let's talk about what will happen. We told you about your structures here to increase surface area. What's going to happen in here with digestion and everything? Well, it makes its own juice, its own fluid. It makes 1 to 2 liters a day, so it makes a lot of it. It's mostly water, has mucus in it. Happens to be slightly alkaline in consistency. So what's happened is you release highly acidic chyme from the stomach. It goes into the duodenum of the small intestine. And right there, it has ducts depositing pancreatic buffers on it. So that brings down the acidity. In addition, now it's mixing with intestinal juice from the small intestine, and that is going to bring it down even further, and in fact, it's going to make it slightly alkaline. So it actually raises our pH above 7. And you will be producing these brush border enzymes in here. So let's take a look then at what's actually going to happen with the chemical digestion. Remember, we've got an added layer of complexity because the small intestine makes its own brush border enzymes, but now we've got enzymes coming from the pancreas as well. So in the end, if you summarize all that up, this is what you get. Carbohydrates, we have both pancreatic and brush border enzymes working on carbohydrates. Note, just a reminder that we got to break them down to monosaccharides. Proteins, we have both pancreatic and brush border enzymes working on those as well. Lipids, only pancreatic, we don't have any brush border enzymes. So your small intestine cannot produce lipid digesting enzymes. We're depending on the ones from the pancreas. Nucleic acids, we got both kinds again.